Hello everyone and welcome to today's session. Uh, Today we're not gonna study anything new. Some of you have texted me regarding the revision of the protein, membrane protein part. So I thought that I'll probably um, just revisit all the things till now what we have studied in the protein part of the membrane. So we're gonna do that, and also I guess in the last class, the video recording for the last class, there is no audio for the YouTube videos which I show for references, right? So I might revisit them again so that we can have a better uh, overall overview of membrane proteins. So we're gonna start in 5 minutes as usual so that more people can join in. Okay. I guess we can start. So basically, membrane has majorly two components, right? Lipids and proteins. Lipids we have covered in quite extensive way, and then we switch to proteins. So for membrane proteins specifically, we add, just like in membrane lipids, we studied about the structure of the lipids, right? Different types of lipid, classification of lipid, and all that. Similarly, for membrane proteins, before going into the type of membrane proteins and all the functions associated with the membrane protein, it is important to know what proteins are at the end of the day, right? So, for that, we started our discussion with amino acids. So, proteins are polymers of amino acids, with each amino acid residue joined to its neighbor by a specific type of covalent bond, which we know is called a peptide bond. Okay, proteins can be broken down to their constituent amino acid by a variety of methods, and the earliest studies of proteins naturally focused on the free amino acid derived from them. 20 different amino acids are commonly found in proteins. Amino acids share common structural feature. All 20 of the common amino acids are amino acids. They have carboxylic group and an amino group bonded to the same carbon atom. They differ from each other in their side chains or R group, which vary in structure, size and electric charge, and which influence the solubility of the amino acid in water. This is the basic representation of amino acid. As you can see, in center there is C alpha. Via C alpha we have 4 different groups. First one is a carboxylic group, then we have amino group, hence amino acid, the name comes from here, and we have H, and then we have this variant R group. Okay, now we'll see how to classify amino acids. So we spent quite a lot of time in understanding the classification of amino acid. So you can classify anything based upon criterias, right? Criteria or parameters. So whichever classification which we are studying, it doesn't mean that you can directly go and say, as you can see there are 5 blocks, right? So if someone asks you that how many different types of amino acids are there, and then you say 5, this answer will be wrong, because they haven't told you on what basis you want us to classify the amino acid, right? So this particular classification which you are seeing on your screen is based upon the R group or the side chain. Okay, so if we classify amino acid based on their side chain, there are 5 different groups of such amino acid, or we can make 5 such groups. The first one is called a non-polar aliphatic R group. The second one is the aromatic R group, then polar uncharged R group, then positively charged R group, and then negatively charged R group. Okay, and again, one more important thing, all these positive and negative charge, they will vary depending upon the surrounding. So this particular classification is at pH 7, okay, physiological temperature and pH. So how many different types of amino acids based upon the R group at physiological conditions? There are 5 different groups. There will be non-polar aliphatic R group, there will be aromatic R group, there will be polar uncharged R group, there will be positively charged R group, and then there will be negatively charged R group. In non-polar aliphatic R group, there is glycine, alanine, proline, valine, leucine, isoleucine and methionine. Okay, and you can see the highlighted part is the side chain. So the smallest amino acid will be glycine, which has H in place of R group, with the smallest group which you can have to satisfy the valency of a central carbon, right? So in non-polar aliphatic R group amino acids will be glycine, alanine, proline, leucine, isoleucine, valine and methionine. Okay, then in aromatic R group amino acid we have phenylalanine, tyrosine and tryptophan. Okay, then polar uncharged R groups, we have serine, threonine, cysteine, asparagine and glutamine. positively charged amino acids, we have lysine, arginine and histidine. While last, negatively charged R group amino acids, we have aspartate and glutamate. Okay, so one more time, the classification. Why I am putting so much importance to this particular slide is because if you understand this classification, then it's pretty easy for you guys to actually predict the nature of the protein or their net charge or something like this, completely based upon the sequence itself, right? So one should be very clear about the amino acids and their basic structural properties. Okay, so in non-polar aliphatic amino acid, we have glycine, alanine, proline, valine, isoleucine, leucine and methionine. In aromatic amino acid, that is side chains or R group who is aromatic in which are aromatic in nature, they are phenylalanine, tryptophan and tyrosine. In polar but uncharged amino acid or side or R group or side chain, we have serine, threonine, cysteine, asparagine and glutamine. For positively charged amino acid, we have lysine, arginine and histidine. Negatively charged amino acid, we have aspartate and glutamate. Okay, so now we look into these group one by one. So in non-polar aliphatic R groups, the R groups in this class of amino acid are non-polar and hydrophobic. The side chain of alanine, valine, leucine and isoleucine tend to cluster together within proteins, stabilizing protein structure through the hydrophobic effect. Glycine has the simplest structure, although it is most easily grouped with the non-polar amino acids. Its very small side chain makes no real contribution to interactions driven by the hydrophobic effect. Methionine, one of the two sulfur-containing amino acids, has a slightly non-polar thioester group in its side chain. Proline has an aliphatic side chain with a distinctive cyclic structure. The secondary amino, that is imino group of the proline residue, is held in a rigid conformation that reduces the structural flexibility of polypeptide regions containing prolines. Aromatic R groups. Aromatic R groups, phenylalanine, tyrosine and tryptophan, with their aromatic side chains, are relatively non-polar, that is hydrophobic. All can contribute to the hydrophobic effect. The hydroxyl group of tyrosine can form hydrogen bonds, and it is an important functional group in some enzymes. Tyrosine and tryptophan are slightly more polar than the phenylalanine because of the tyrosine hydroxyl group and the nitrogen of the tryptophan indole ring. Tryptophan and tyrosine, and to a much lesser extent phenylalanine, absorb ultraviolet light. This accounts for the characteristic strong absorbance of light by most proteins at a wavelength of 280 nanometer, a property exploited by researchers in the characterization of the proteins. This is the absorption spectra of aromatic amino acids, and you can clearly see that at and around 280 nanometers these absorb light. Okay, except phenylalanine, which shows some absorbance, but tyrosine and tryptophan are showing significant absorbance at 280 nanometer. Polar uncharged R group. Polar uncharged R groups, the R group of these amino acids are more soluble in water or more hydrophilic than those of non-polar amino acids because they contain functional groups that form hydrogen bonds with water. This class of amino acid includes serine, threonine, cysteine, asparagine and glutamine. The polarity of serine and threonine is contributed by their hydroxyl groups, and the polarity of asparagine and glutamine is contributed by their amide groups. Cysteine is an outlier here because its polarity contributed by its sulfur hydryl group is quite modest. Cysteine is a weak acid and can make weak hydrogen bonds with oxygen and nitrogen. Asparagine and glutamine are the amides of two other amino acids also found in proteins, aspartate and glutamate respectively, to which asparagine and glutamine are easily hydrolyzed by acid or base. Cysteine is readily oxidized to form a covalently linked dimeric amino acid called cysteine, in which two cysteine molecules or residues are joined by a disulfide bond. The disulfide linkage residue are strongly hydrophobic, nonpolar. Disulfide bonds play a special role in the structure of many proteins by forming covalent links between parts of polypeptide molecules or between two different polypeptide chains. Positively charged basic R groups and negatively charged acidic R groups. Positively charged R groups, the most hydrophilic R groups are those that are either positively charged or negatively charged. The amino acid in which the R groups have significant positive charge at pH 7 are lysine, which has a second primary amino group at the position on its aliphatic chain, arginine, which has a positively charged guanidinium group, and histidine, which has an aromatic imidazole group. As the only common amino acid having an ionizable side chain with near neutrality, histidine may be positively charged or uncharged at pH 7. Histidine residue facilitate many enzyme catalyzed reactions by serving as proton donors and acceptors. Negatively charged or acidic R groups, the two amino acids having R groups with a net negative charge at pH 7 are aspartate and glutamate, each of which has a second carboxylic group. So the overall summary for the amino acids is the 20 amino acids commonly found as residues in proteins contain an carboxylic group, an amino group, and a distinctive R group substituted on the carbon atom. The carbon atom of all amino acids except glycine is asymmetric, and thus amino acid can exist in at least two stereomeric forms. Only the L stereomers of amino acid with a configuration related to the absolute configuration of the reference molecule L-glyceraldehyde are found in proteins. Amino acids can be classified into 5 types on the basis of the polarity and charge of their R group. Other less common amino acids also occur, either as constituent of proteins, usually through modification of common amino acid, or as free metabolites. Amino acids vary in their acid-base properties and have characteristic titration curves. Monoamino monocarboxylic amino acids with non-ionizable R groups are diprotic acids at low pH and exist in several different ionic forms as the pH is increased. Amino acids with ionizable R group have additional ionic species depending on the pH of the medium and that of the R group. Other way of classifying amino acid, if we classify all the 20 amino acids based on whether they are polar or non-polar, we have first classification. There you will have in non-polar amino acids in non-polar amino acid we have alanine, valine, leucine, isoleucine, proline, methionine, phenylalanine and tryptophan. While in polar we have glycine, serine, threonine, cysteine, glutamine, tyrosine, asparagine, lysine, arginine, histidine, glutamic acid and aspartic acid. If we classify amino acid based on their side chain or the type of R group, we have aliphatic, which contain glycine, alanine, valine, leucine and isoleucine. Then we have aromatic, which has phenylalanine, tyrosine and tryptophan. We have polar uncharged, maybe serine, threonine, asparagine and glutamine. Sulfur containing, we have cysteine and methionine. Cations, we have lysine, arginine and histidine. Anions, we have aspartic acid and glutamic acid. Amino, we have proline. Okay. So peptides and proteins. Two amino acid molecules can be covalently joined through a substituted amide linkage termed as peptide bond to yield a dipeptide. Such a linkage is formed by removal of the elements of water, dehydration, as you can clearly see here. A hydroxyl moiety from the carboxylic group of one amino acid and a hydrogen atom of the amino group of another amino acid. When a few amino acids are joined in this fashion, the structure is called an oligopeptide. When many amino acids are joined, the product is called a polypeptide. Proteins may have thousands of amino acid residues. Although the term protein and polypeptide are sometimes used interchangeably, molecules referred to as polypeptides generally have molecular weight below 10,000 Dalton, and those called proteins have higher molecular weight. No generalizations can be made about the molecular weight of biologically active peptides and proteins in relation to their function. To prove the last statement, you can clearly see that there is no particular relation with the molecular weight and their function. So cytochrome C protein has 12,400 Dalton, number of residues is 104 and the number of polypeptide chain is 1. On the other hand, chymotrypsin, it has 25,000 around molecular weight, has 241 residues and has 3 polypeptide chains. Hemoglobin has 64,500 molecular weight, Dalton molecular weight, has 574 residues and is divided into is made up of 4 polypeptide chains. Amino acids can be joined covalently through peptide bonds to form peptides and proteins. Cells generally contain thousands of different proteins, each with a different biological activity. The ionization behavior of peptides reflects their ionizable side chain as well as the terminal amino and carboxylic group. Proteins can be very long polypeptide chains of 100 to several thousand amino acid residues. However, some naturally occurring peptides have only a few amino acid residue. Some proteins are composed of several non-covalently associated polypeptide chains called subunits. Simple protein yield only amino acid on hydrolysis. Conjugated proteins contain an additional some other components such as a metal or organic prosthetic groups. In protein structure, in a protein's three-dimensional structure, the more hydrophobic amino acid should reside. Amino acids should the more hydrophobic amino acid should reside together, forcing that portion of the protein into the membrane, while the more hydrophilic amino acid should locate into protein's domain that will interact favorably with water. Therefore, membrane proteins analogous to membrane lipid must also be amphipathic, having both hydrophobic and hydrophilic regions. The outstanding question is how the amino acid sequence relates to a protein's location, either into or out of the membrane. One direct possibility would be to determine the precise location of each amino acid in a protein by X-ray crystallography. Unfortunately, this has proven to be very difficult task for most membrane proteins. There are now more than 30,000 known water-soluble globular proteins and more than 20,000 membrane proteins. However, as it is much easier to obtain crystal from globular proteins, more than 6,000 crystallographic structures of these proteins have been determined compared to few dozen structures of integral membrane proteins. So, are there other methods to identify protein domains that prefer a location in the hydrophobic core of a membrane? To answer this question, the amino acid cannot simply be lumped into classes but instead must be characterized individually. this approach involves hydropathy plot. So we'll understand a hydropathy plot now. Why there is no volume?
And specifically what you need in order to solve a problem that you might encounter on an exam. So what exactly is a hydropathy? So today we're going to talk about all things hydropathy plots, and specifically what you need in order to solve a problem that you might encounter on an exam. So what exactly is a hydropathy plot? Well, a hydropathy plot is going to measure our hydropathy index, and it's going to measure the number of amino acid residues in a singular protein. What a hydropathy index does is it measures our free energy change associated with the movement of amino acid side chains from a hydrophobic environment to a hydrophilic environment. So when you see these graphs, you should immediately start to think of transmembrane protein. This is what a transmembrane protein looks like. Notice how it spans across the entire membrane. The amino acids inside the membrane are going to be hydrophobic, and the amino acids found outside of the membrane are going to be hydrophilic. So it's going to take about 20 amino acids in order to cross our membrane. So let's think about how you would solve a problem like this. First, we're going to need to know how to read our hydropathy plot. Make sure we understand that our positive free energy values are going to be associated with our hydrophobic amino acids, aka our amino acids found inside of the membrane. These are our negative hydropathy values. These are going to be associated with our hydrophilic amino acids, and they'll be found outside of our membrane. Let's take a look at an example of a protein. Notice how this protein crosses our membrane once, so it's a transmembrane protein. Since it crosses once, we can see that our hydropathy plot would look something like this. Notice how there is one positive peak. Again, notice how it crosses once. Let's say that our N-terminus is located on the outside or extracellular membrane or luminal face of the membrane. These are all different ways to say outside of the membrane. We would know that our C-terminus would have to be found on the inner membrane, or you could hear the words inner leaflet of the membrane. These mean the same thing. So let's look at a hydropathy plot. Notice how there are 4 positive peaks in our graph. So we're looking at a protein that's going to cross our membrane 4 different times. Now let's consider a question that could be asked based on this hydropathy plot. Consider the hydropathy plot for a transmembrane protein with its amino terminus located on the inner leaflet of the plasma membrane. Where is its carboxy terminus located? So let's walk through how I would solve this problem. So I'm going to set up my hydropathy plot here based on the question that was asked. I was told that the amino terminus is on the inner leaflet, so I will draw it like so. Now I know for each positive spike in my graph, I am crossing the membrane to another side. So for my first cross, if I start on the inside, I am going to end up on the outside. For my second cross, I come back into the inside. So for my third cross, we head back to the outer leaflet, and for the fourth cross, we know that our carboxy terminus must be in the inner leaflet of the membrane since our protein ends. If that didn't make sense, take a look at this protein and notice the 4 transmembrane segments. This protein is exactly what we just demonstrated from our hydropathy plot, so make sure that this makes sense to you, and if not, try to go back and redo the question. I encourage you to quiz yourself on these problems to make sure that you aren't just blatantly following along. For example, you should be able to answer the question, what would happen if our protein had only 3 transmembrane segments? Or what would happen if our carboxy terminus was located on the outer leaflet of the membrane? And what kind of amino acids would be found there? These are the kind of questions you should be able to answer in order to master the concept. As usual, I encourage you to join the Campus Wire and go to office hours. Dr. Alleman also has an additional video on how
So that was the hydropathy index. By this method, each amino acid is assigned a unique hydropathy index value that expresses its lipid water partition. These values represent a measure of the free energy change accompanying the movement of an amino acid from a non-polar solvent to water. Probably the most used approach for this type of membrane study came from Kite and Doolittle in 1982, and this is the Kite and Doolittle hydropathy index profile. So by Kite and Doolittle, most of the non-polar amino acids have a positive index, while most of the polar amino acids have a negative index. Once a hydropathy index of the amino acid is established, the next step is to plot the index values for each amino acid in the protein's sequence from N terminal to C terminal, and we have visited this and saw that the hydropathy or the hydrophobic patch which we are seeing in the hydropathy plot is in sync with the glycophorin protein and the residues which are in the membrane. Okay, then after classification of proteins, we try to understand how many membrane proteins types are there. So there are peripheral protein, amphitrophic protein, integral protein, and lipid linked protein. And from here onwards, I guess I'll start the videos.
In this video, we're going to explore membrane proteins. Did you know that the cell membrane can be composed up to 75% protein? So most cell membranes have about 50% or less protein. And proteins are there because the cell membrane uses proteins for pretty much everything that it does, all of these cell membrane processes that it performs. So just to remind us what a cell membrane actually is, a cell membrane is made up of little things that look like this, which are called phospholipids, and they come together and form what we call a lipid bilayer. So over here, I've pre-drawn a lipid bilayer, and it'll look something like this. It'll be made up of a lot of these small phospholipids that we've drawn above, and it'll make up our bilayer. So you can see that there are two layers of these phospholipids. Now, there's two major types of proteins in the cell membrane. The first can look something like this. And this can appear anywhere in the cell membrane, and there are usually quite a few of these throughout the entire cell. So this is what we call an integral protein. You'll notice that it's called an integral protein because you can think of it like it's integrated throughout the entire membrane. Another type of protein that we might encounter might appear on top of the membrane. Occasionally, it might be slightly into the membrane, and it can also rest on top of integral proteins. And this we call peripheral proteins. And the reason why we call it a peripheral protein is because it's on the peripheral or the outside of the cell membrane. The difference between peripheral and integral proteins is that integral proteins are really stuck inside the cell membrane. As you can see in this picture, the integral protein is really inside the membrane, and as a result, it'll be very difficult to remove. Peripheral proteins kind of attach and remove themselves from the cell membrane or from other proteins. They generally are there for different cell processes. So for example, a hormone might be a peripheral protein, and it might attach to the cell, make the cell do something, and then leave. Peripheral proteins can also exist inside the cell on the cell membrane. Another type of protein is extremely rare, and it can appear inside the cell membrane like that. And we call this a lipid-bound protein. Why might you think a lipid-bound protein is so difficult to find, so rare? Well, the reason why is because proteins are there to interact with the outside environment. And lipid-bound proteins are stuck on the interior of the cell membrane itself. So it can't really interact with the outside of the cell or the inside of the cell. So it doesn't really serve a big function in terms of the cell membrane performing its duties. We're going to spend a little bit of time talking about two types of integral proteins that are extremely important because these two proteins are found all over the cell and they help the cell maintain homeostasis or balance. The first type can look something like this. Again, this is an integral protein. What do you think this protein might be used for? This isn't two proteins, it's actually one protein with a hole through it. Well, this protein is actually used to allow things to pass through the cell. We call this a channel protein. And like the name kind of implies, there's a channel or hole inside the protein that lets things pass through. So for example, if there is some sort of ion, let's say this is an Na plus ion, a sodium ion, this is outside the cell. And the cell at this point really needs these sodium ions to perform a really important process. So what the channel proteins do is they'll allow these outside extracellular ions into the cell. And normally, these sodium ions wouldn't be able to pass through the cell membrane just by themselves. These channel proteins allow our bodies to take in different materials from the outside environment into our cells. What they can also do is they can also do the reverse. So let's say your cell has way too much sodium and it needs to get rid of it. So channel proteins can start pumping these out. Channel proteins generally don't require energy, so there's no energy needed. Sometimes we call energy ATP. And another thing that's special about channel proteins is you'll notice that it'll go with the concentration gradient. So out here there's a lot, and inside there's very little. So it'll pump from where there's a lot of sodium into where there's very little. So it'll go what we call down a concentration gradient. Concentration gradient. The second type of very important integral protein is called a carrier protein. And like the name implies, it carries substances into the cell. I kind of picture it like a baseball glove, like this. So if there's a molecule that's outside the cell, and the cell actually needs this molecule, so what the carrier protein will do is it'll actually protect this substance so that it can enter the cell safely. It can also do this in reverse. It can take something inside the cell and pump it outside the cell. And this type of protein is really important because unlike channel proteins, carrier proteins can go against the concentration gradient. And this is really important because say your cell has a lot of chloride ions and your body needs more to perform a certain process. So what your body can do is it can bring more chloride ions into your cell even though your cell already has a lot of chloride ions. So carrier proteins can sometimes use energy or ATP. Finally, there's a type of protein that can exist on any of these that we've drawn here. And this is what we call a glycoprotein. So what a glycoprotein would look like is there will be a chain of sugars attached to a protein. And it can be on integral proteins, peripheral proteins, channel proteins. Glycoproteins, you'll notice have the prefix glyco, which means sugar. And basically it's just sugar plus protein. And the purpose of glycoproteins is that it's used in signaling. So it allows a cell to recognize another cell. So in summary, in this picture that we have drawn out of a cell membrane and several different proteins,
that was the basic introduction to type of proteins, and then we have about the peripheral proteins. So peripheral proteins are held to the membrane surface primarily through electrostatic or hydrogen bonds. They can be divided into two basic types: those that are attached through charges on the surface of integral protein, and those that are attached to the anionic head groups of phospholipids. Peripheral proteins can be removed easily and cleanly from the surface of membranes by either altering the pH or the media salt concentration. By definition, a true peripheral protein exhibits little or no interaction with the membrane hydrocarbon interior and is removed from the membrane without attached lipids. Classic examples of peripheral protein include cytochrome C and the myelin-basic proteins. Amphitrophic proteins. Amphitrophic proteins are a relatively new class whose importance is rapidly becoming more apparent. Unfortunately, there is a lot of overlap between the peripheral proteins that are bound to the membrane surface phospholipids, integral, ecto, and endo proteins, and amphitrophic proteins. Amphitrophic proteins can alternate between conformations that are either water soluble or weakly bound to the membrane surface through a combination of electrostatic and hydrophobic forces. Since the membrane interaction is reversible, an amphitrophic protein coexists as both a free globular protein and a membrane bound protein. The water soluble globular form is converted into the membrane bound form following a conformation change induced by phosphorylation, acylation, or ligand binding that exposes a previously inaccessible membrane binding site on the protein. Membrane interaction is often based on production of a hydrophobic loop of amino acids, sometimes an alpha helix that can penetrate into the lipid bilayer. Integral protein. It is estimated that 20 to 30% of a cellular proteins are integral, and these proteins are tightly and permanently bound to the membrane. While peripheral and amphitrophic proteins are weakly bound to the surface of membrane and can be readily dissociated, producing lipid-free globular proteins, integral proteins are integrated into the membrane hydrophobic interior and can only be removed by more drastic means. Historically, integral proteins have been separated from membrane through acetone and other nonpolar organic solvent extractions, chemotropic agents, or detergents. Although thousands of globular proteins have been crystallized and their 3D structures accurately resolved by crystallography, only a few dozen integral membrane proteins have been resolved. The problem of obtaining 3D structures for the integral protein is in obtaining pure crystals in the first place. While peripheral proteins are obtained as monomers that are free of membrane lipids, integral proteins are isolated as aggregates with lipids still attached. The aggregates are almost impossible to crystallize. Therefore, structural information about integral protein is often obtained by indirect methods like enzyme degradation studies and hydropathy plots. Endo and ectoproteins. Endo and ectoproteins do not traverse the entire membrane. Instead, they are inserted into the membrane's hydrophobic interior from either the cytoplasmic side, endo, or the extracellular side, the ectoprotein. An example of such protein is cytochrome B5. Then we studied about transmembrane proteins. A transmembrane protein must span the entire membrane with segments exposed on both the outside and the inside aqueous spaces. The membrane that must be spanned is composed of a lipid bilayer that is divided into three sections. The inner hydrocarbon region is approximately 2.7 to 3.2 nanometer thick. The very narrow boundary region between the hydrophobic inner core and the hydrophilic interfacial region is approximately 3 angstrom. Finally, the polar head group region is approximately 8 to 10 angstrom, although this may be wider in membranes that include large amount of carbohydrate-rich components. All membrane proteins are 100% asymmetrically distributed with respect to the membrane and do not flip-flop across the membrane. Therefore, transmembrane proteins are locked in place. A characteristic of many transmembrane proteins is the presence of tyrosine and tryptophan at the aqueous interface. These amino acids serve as interfacial anchor that can interact simultaneously with the membrane hydrophobic interior and the aqueous exterior. Also, for reasons not entirely clear, the cationic amino acid lysine, histidine, and arginine occur more commonly on the cytoplasmic side of the protein. This is referred to as positive inside route. Since each amino acid In an alpha helix span approximately 1.5 angstrom, it takes 20 amino acids to cross the hydrophobic interior. Is expected that As expected, the transmembrane alpha helices are generally devoid of polar amino acid, while non-membrane spanning portion of the protein are usually enriched in these amino acids. Alpha helices can cross the membrane a single time or multiple times. So this is all what we have discussed till now with respect to the classification of proteins. So in next classes we're gonna build more upon this only. Thank you for your time, and if you have any queries, feel free to write to me. Thank you.